Hey everyone, sorry we have to do it this way today. Hopefully we can get back in our classroom on Thursday, so we'll rather be safe than sorry, but we're just going to start chapter 3 right now. So these are some key characteristics of a reliable microscope. So you have magnification, which looks at the ability to enlarge objects in appearance. And then we have resolution, so the capacity to distinguish or separate two adjacent objects. I'd also like to point out the PowerPoint that I put online is apparently the students' PowerPoint, so there are some things missing. So you'll have to be on the lookout. I know resolution wasn't filled in. I had to go back in and refill that in. So just be on your P's and Q's while you're watching this, because I don't remember everything that was that I needed to fill in. So magnification in most microscopes results from an interaction between the visible light waves and the curvature of the lens. The extent of enlargement is considered the magnification. So you can see right here, we have sizes of these things. So here's a human hair, 200 times zoomed in, or 200 times magnification. And then you can see that they've taken a subset of this picture, and this is pollen at 200 times, or 2,000 times, excuse me, magnified. You are not able to see this on the compound microscopes that we have in our classroom. Our compound microscopes only go up to 1,000 magnification, and that's when we use that immersion oil. So we would never be able to see this in class. It would look like just a dot. Now you can see, I probably can't see my cursor. So you have the pollen right here. A lymphocyte is one of your blood cells. We have yeast. We have a red blood cell right here. E. coli, Ebola, which we looked at last week. You can see the tiny little rhinoviruses right there. Staphylococcus. So I wanted to point out this right here, that means micrometers. So when you see that it means micrometers. When you see NM, that means nanometers. All right. So looking at the size of things, this is a nail right here that you can see it magnified. Doesn't tell you how much it's magnified, but they magnified it, and you can see that there's bacteria all on the head of that nail, just to show you how small these organisms can be. So again, these are the size of things. So here's a typical eukaryotic cell. That's why when we were looking under the microscope, you were able to see the fungus, the algae, all those, because they are eukaryotic. So they typically are very big. Here is your nucleus. Prokaryotic cells are smaller. That's why when you were looking at bacteria cells, a lot of you were like, am I supposed to just be looking at those purple dots? That's because we weren't zoomed in pretty far. Even on our compound microscopes, you can't really see very much. They're still going to just look like purple dots. And then a virus, we would never be able to see those on our microscopes because they are very, very microscopic. So we have some bacteria right here just chilling. Let's see. So a red icon, you can see the ones right here. That means that they are artificially colored. With your microscopes, the slides that we looked at the other day, most of them were stained, so that you could see that. If you don't look at them stained, it just looks clear. So a lot of things are stained, and these are all not microscopically, sorry. These are all stained or artificially colorized, so that you can see the difference between their background and the actual thing you're supposed to be looking at. But you can see by size. So here's a tick. I'm sure we've all seen a tick in real life. They're pretty small, but not nearly as small as red blood cells. We would be able to see red blood cells on the microscopes that we have in the lab. I don't know if we're going to look at them this year. That would be for a later time, though. And then we got E. coli, viruses, and then the smallest thing that we would have to use an atomic force microscope for would be the DNA double helix. You would probably, I've never seen it in a microscope. I've only seen it in pictures. But with scanning electron microscopes, you'd be able to see all of these, and we'll get into the different types of microscopes. I believe we'll get into them tonight. So we have units of measurements. Like I said earlier, the UM is micrometers and NM is nanometers. And you can see all the units of measurements right here. I have already created your Unit 1 test. And there will be a couple of questions asking you to convert the micrometers to nanometers and things like that. So this is an important slide to hold on to. We talked about this last week with Leeuwenhoek's microscope. So this is just a refresher. So you have your lens right here, and you put the specimen that you're looking at on the tip of that pen. Then you can move everything up and down based upon the screws going on. Now this is more of a representation of what you have in the classroom. There will be questions on your test about the different parts of a microscope. And I wish we were in class so I can show you. I might spend some time on Thursday showing you in person what the microscope looks like and the different parts, but hopefully this picture will be good enough. But right here, what you actually look into, that's what we call the lenses, or the ocular lenses. We call them eyepieces. They tend to have a 10 times magnification on them. Right here, the part that kind of swivels a little bit, if you saw me come up to you and kind of swivel the eyepieces over, that's where the body tube is held. So that actually transmits the image from the objective lens to the ocular lens. So the objective lens is down here. The arm is where you are supposed to put one hand when you're holding a microscope, as well as the base. So you're supposed to put your hand on the base and the arm when you tote it around. The objective lenses are the swiveling pieces that you that have different magnifications on them. We call them objective lenses. You have the stage, which actually holds the microscope in place. The condenser, which helps focus light. The diaphragm, which controls the amount of light, is right underneath the condenser. You have your light source. Sometimes they'll call it an illuminator. On our microscopes, excuse me, my dog saw his reflection. But some microscopes, these are a little bit higher up, like the ones in our classroom. So you have the coarse focusing knob right here. The coarse knob is what is going to zoom in very quickly. And then you have your fine focusing knob. So once you actually get any kind of image, we're using the coarse focusing knob, then you use this fine focusing knob to kind of make that image a little bit clearer. There will also be a question on your test about the path of light. So the base, and I don't think it starts that early on, but the light does come out here, comes through the illuminator, through the condenser lenses, through the specimen or the slide that's up there, goes up through the objective lenses, goes through the body tube into the prism, and then out the ocular lenses where you look through. So total magnification, we talked about this a little bit in lab. You have to multiply the objective power and the ocular power. So you have to multiply the piece that you look through, the eyepiece, with the objective lenses, so the tubes that are on the microscope to zoom in. And that's how you get total magnification. You have to multiply those two things together. So at your highest magnification, when we talk about using the immersion oil, on the ocular lens, you can see it only says 100 times, but because you're looking through the eyepieces that are 10 times, you have to multiply 10 times 100 to get 1,000. And that is your total magnification when you're looking in there. These are just going to be different types of microscopes. So this was in 1775. You can see it kind of looks like a magnifying glass. So he would put something on the tip of this pen and look at it through the magnifying glass microscope kind of thing. This was in 1765, so this was a chest microscope. You have all these little pieces chilling right here, and he would put a sample on this part right here, and you would look out of it from this part up here. This was a toy microscope in 1765, so again, you would put the specimen right here. This would bring in the light, so you would point that like towards the sun, the light would go up, and you would look out or look into it right here. A solar microscope, so we've got, you put the specimen out here, it brings in the light, and you look at it right here. All right, so looking at resolution, so the capacity to distinguish or separate two adjacent objects and depends on the wavelength of light that forms the image along with the characteristics of the objective. So with really big wavelengths, you're not able to see as much. That's why you have to zoom in and out and do the condenser and things like that. You have to shorten out that wavelength in order for you to get a more clear picture. Because with the wavelengths being so big, they're not able to get in those tight spaces. And so you get a blurry image. So we have to tune in that light, make the wavelength shorter so they're able to get in these tight spaces and have a clear image. So this is just looking at the same thing. So when the wavelengths are too large, they're not able to get into tight spaces and you get a fuzzy, undetailed picture. But when you shorten out those wavelengths, you do tend to get a clearer image because it's able to get into those nooks and crannies. And I explained to a couple of you one-on-one as I was going around the lab the other day about why we have to use the immersion oil. Because you can just use the 100 times octave normally. It tends to get really fuzzy. It's not as clear if you use the oil, but you can do it. The reason it gets fuzzy is because there's not a lot of distance in between here. And so our light immediately fragments away. And if you're further away from the slide, it's okay because it'll fragment just a little bit and you'll be able to pick it up still because you're so far away. But because you're really, really close when you use that 100x lens, the light fragments. This oil just kind of prevents it from going too far, so it's able to be picked up by that lens. So getting into electron microscopy, so it uses beams of electrons instead of light. The shorter wavelength of electrons gives greater resolution, and it's used for images too small to be seen with light microscopes, which light microscopes are what you use, such as bacteria and viruses. There's two different types of electron microscopes. The first type that we're going to talk about is transmission electron microscopy. So a beam of electrons passes through ultra-thin sections of a specimen through electromagnetic lens, then focused on a projector lens. Specimens may be stained with heavy metal salts for contrast. Those heavy metal salts tend to be uranium, molybdenum, tungsten, and lead. So these heavy ions readily react with the electron beam and produce a very high contrast so you're able to see better. This magnifies objects 10,000 to 100,000 times. Again, on our microscopes, you're only able to get to 1,000 times. So it gets very, very detailed and very, very small. images. So you can see right here what they look like. Again, whenever you see this red thing right here, that just means that it was artificially colorized. And that's saying it was colorized with the transmission electron microscope. So those heavy salts and it reacting differently based on how close or how far away these objects are depends on the color it's going to be when it shows up through the viewing eyepiece. It doesn't mean that they did this with a computer program, it's just how it goes through with the electron microscope. And you can see here it talks about, so we have an electric gun and the beam comes down. Your specimen is right here, so we have the electromagnetic condenser lenses coming through right here. It goes through the specimen, through the electromagnetic objective lenses, and through the projector to get into your eyesight. The second type of electron microscope that we're going to talk about is the scanning electron microscope. So an electron gun produces a beam of electrons that scans the surface of an entire specimen. Secondary electrons emitted from the specimen produce a three-dimensional image. So you can see this is what a scanning microscope looks like. So we have an electron gun up here. It goes through the electron beam and then the electromagnetic lenses. We have a viewing screen right here. And this is what collects the electrons. It is amplified and it comes through here. And the electrons send out through here, and this is where it comes for the viewing screen so that you can see it. And again, these are artificially colored, not on purpose, but because that is what the electron microscope shows. So like I said, there's two types of electron microscopes. The transmission transmit electrons through the specimen. Darker areas represent thicker, denser parts, and lighter areas indicate more transparent, less dense parts. And the scanning electron microscope provides detailed three-dimensional view. SEM bombards surface of a whole metal-coated specimen with electrons while scanning back and forth over it. So the big takeaway from this slide, which you will be asked on your test, is what the difference is. I believe it's going to be an essay question, just FYI. But transmission goes through the specimen, while scanning goes on the surface. So here are just more pictures. So here's a light microscope. So this is what you would be able to see in your class. And then this is a scanning electron microscope. You can just see the difference in how we're able to see things better and how it looks a little bit better. Okay, so wants me to go over the advantages and disadvantages of each. So talking about light microscopes, the advantages is that it's cheaper, which is why most colleges have these. You can look at the specimen while it's alive. It is good for cells, but not organelle detail. So you can see the cell, you just can't notice anything that's happening on the inside. Some disadvantages is it's a poor depth of field. You can't get much magnification out of it. The whole image isn't as sharp. So that's for the light microscope. Now some advantages for the electron microscopes. You can see very well. They have a resolution of 2 nanometers compared to 2 micrometers that we had for the light microscope. Disadvantages of electron microscopes is that they are very costly. They can cost thousands of dollars. And because we're basically dosing these microbes with electrons, they do kill the specimen to be able to view it. And the specimen preparation is very evolved. So you have to kill it, fix it, and coat it with metals, which increase image contrast. So they have to kill it beforehand. I apologize, I said it wrong earlier. But they kill it beforehand just to make sure it doesn't move, because you would hate to spend these thousands of dollars using this machine and it moves and you don't get a good image of it. So they kill it and fix it to the slide, and we'll talk more about what fixing means to the slide. I believe on Thursday we'll get into that. And then again, this is just the different electron microscopes. You have your transmission over here, and you have your scanning over here. And so it just shows you how each one would look depending on which one you used. Alright, so here's a section through of some kind of protozoa. Toxoplasma gondii, so this causes toxoplasmosis. And you can actually see the organelles chilling here. So this should be a ribosome right here. You have the nucleus right here. Mitochondria. Believe the GO would probably be the Golgi. Cell membrane. Let's see, it tells me what all of them are. CE, where's CE? Here's our centrioles, which help with cell division. And then these DG and AMs, those are our granules. All right, preparing slides. So we will get into it tonight. So we can prepare fresh live preparations so that you see live cells whenever you look in the microscope. Whatever you saw last week, those were all dead and have been fixed to the slide. But at some point this semester, you will have experience with creating wet mounts and hanging drop mounts. So it allows you to actually look at characteristics of living cells to see what they look like when they're actually living. So their size, how they move, their shape, arrangements. You also will have fixed mounts, which are what we looked at last week and what we would have looked at on Tuesday had we had class, are made by drying and heating a film of specimen. The smear is stained using dyes to permit visualization of cells or cell parts. So that's like that Staphylococcus that we looked at. It was stained purple. They are not normally purple. They're normally a goldish color. But in order for us to be able to see them, they changed them to, they dyed them purple. What is this? You performed a wet mount when you looked at live cultures. I had some live cultures prepared that you could have looked at the other day, but I didn't pull them out. Just because I know y'all were getting used to the microscopes. If we would have had class tomorrow, I would have let some people use some wet mounts, but because we don't have class, we'll get some practice with it later. So again, this is how you prepare a wet mount. So you put your dropper, normally we use like pond water when we look at this. So you just put a drop of it on there, and with your cover slip, I've never had this fancy little tool. I don't know what that's supposed to do, but I normally just drop the cover slip down very gently. You can also do it where you put Vaseline and make like a cool little like moats kind of thing where you can keep the cover slip from damaging anything underneath it. So you make a like square of Vaseline, put your drop in there, and then you put the cover slip on top of the Vaseline so that you don't damage anything, because sometimes dropping these cover slips, if you do it too harshly, you can kill whatever's down there. And here's a hanging drop. The hanging drop is kind of what I just explained, except for my college was too poor to buy the depressions slides. But you can see it's got a little divot right here, and you put the cover slip on top of it. And you just have like the drop kind of hanging down. So you can see it hanging down right there. All right. So different types of staining. You will be able to stain in this class at some point. In the next coming weeks, we'll probably get into staining in the lab. And again, that's just colorizing microorganisms with the dye so that we're able to see them better. A smear is a thin film of material containing microorganisms spread over the slide. Most of the slides that you looked at were smears. They just kind of spread it across and then put a cover slip on top of it. Microorganisms are fixed to the slide, which kill the microorganisms. And they normally use, well, from my experience, they normally use heat to do that. Live and or unstained specimen have little contrast with the surrounding medium, and live specimen are used to study cell behavior. Live specimen are sometimes a lot more hard, not a lot more hard, are harder to look at because they are moving, so you have to continually move the stage around to see them better. But like I said, we'll get into that at some point. So here's fixed, and you will do a fixed stained smear at some point. That's why you have those inoculating loops in your drawers. So you just put it on there. This is for solid and this is for liquids, but it's pretty much the same thing. You just rub that loop across with the specimens on the end of it. And then you heat fix it. And I love this slide because it says use a slide holder, not your hand, but then they literally have a picture of a guy using his hand. Don't understand it. But you will use not Q-tips, Lord Jesus. You use clothespins. So you use a clothespin on your slide and hold it over the flame, which is why you also have access to the Bunsen burners. So we will use the Bunsen burners whenever we fix stains, fix microorganisms onto slides. All right, so we're going to talk about positive and negative staining as well. We'll get into the kind of logistics of that. They're a little bit different. But positive staining is colored by dye. They're not stained, they're generally white. The basic dyes, they normally have the color that they're going to change it to in the name. So we have crystal violet, methylene blue, which is the one that I'm most common with or most used to using is methylene blue. It changes everything blue. Saffron, methylite green, and there's different types of these positive stains. We have simple stains, differential stains, and special stains. And we'll get into all of those more in depth in just a second. And then you have negative stains. So they're dark gray or black. They tend to be acidic. So we have necrosin and Indian ink. These are to see capsules or spores. And here's a negative stain of oral bacteria. So bacteria that's been in your mouth. So yeah, like I said, we're going to get into these now. So subtypes of positive stains. We have simple, which is the use of one type of agent or dye. The common dyes for simple stains include methylene blue, basic fusion, carbonyl fusion, crystal violet, methylene green, and saffron. Differential stains uses two types, primary and a counter stain, to differentiate between two cell types or cell parts, such as spores and the parent vegetation cells that produced it. So we'll use two different types of dyes just so that you can see different things going on. And examples of those are gram staining and acid-fast stains. So gram staining, it classifies bacteria into gram positive or gram negative. A couple of you asked me the other day, what does this gram positive mean? I told you we would get into it later, but it's just a type of bacteria. So now we're going to get into it. So gram positive bacteria have a thick peptidoglycan cell wall, while gram negative have a thin peptidoglycan cell wall and a layer of lipopolysaccharides. So you can see right here we have our gram negatives. Those tend to be dyed purple. And you have your gram-positive, which tend to be dyed blue. We have our acid-fast stains. So it binds only to bacteria that have a waxy material in their cell walls, which is not decolorized by acid or alcohol. So we have those for Mycobacterium, which is a genus of pathogen that causes diseases such as tuberculosis and leprosy. And we have Nocardia, which is a disease found in water and soil that can affect the mind and lungs. So acid-fast staining, you can see we got some going right here. This is from blue lung tissue. So this is Mycocardium. So Mycobacterium, so this is probably tuberculosis, which it might say at the top, but I have my notes covering it. Yep, this is tuberculosis. All right, special stains. So you use this to distinguish parts of microorganisms. So you have the capsule stain, the endospore, and the flagella stain. The capsules are gelatinous covering that do not accept most dyes. So that's why we have to use a negative staining for this. Suspension of India ink and necrosin contrast the background of the capsule, which appears like a halo around the cell. So you can see we've got little like halos going on around here. This again is negative staining, and you can see the capsules going around it. Endospores are resistant dormant structures inside of some cells that cannot be stained by ordinary methods. We use a primary stain of Machelite green, usually with heat. It decolorizes cells with the water. A counter stain, a stain that with coloring contrast to the principal stain. So spores appear green within red or pink cells. So you can see the endospores right here, this little blue line going on right here. And then flagella staining. So flagellas are structures for movement. Uses a mordant in carbonyl fusion. And you can see we got all these flagella right here. All right in there. And again, we have flagella. All right, so that's end of the class for today. Remember, your chapter 1 homework is due tonight, or depending on when you watch it, it's due on August 31st by midnight. And then the chapter 3 homework and quiz will open August 31st at 12:01 a.m. So probably by the time you watch this, it will be open, and it will be due by September 7th at midnight. You also, I'll go ahead and tell you when your exam is, since I've brought it up a couple times. Your exam will be September 14th. I am going to let you take it online. It'll open on the 12th, which is a Sunday at 12:01 a.m. And then it will be due on that Tuesday by 11:59 p.m. We will not have the lecture that night. You'll still have lab, so you'll have to come to lab. But you will not have the lecture, so that you will have plenty of time to take the exam. Let me know if you have any questions. Bye.